Hello and welcome back again to Study with RK. Hope you all are doing well. Here I am back with another guest lecture session. Yes, you already have got the idea after seeing the title of the video. This lecture is going to be about cell cycle and that too in specific content oriented. So what happens many a time that the concept of the students is clear but they cannot execute the same during exams. So with that thought process, I decided to make this content oriented so that it becomes easy to write the answer from this video itself. So now you must probably be aware about cell division. But before cell division, there are few processes or say preparations which is must for the cells to divide. Now what kind of preparations? So preparations such as growth of cell will occur, then collection of several raw materials before division, etc. So all these things will sum up and form a regular cell cycle. Now what do you mean by cell cycle? So cell cycle is a series of events that take place in a cell which results in DNA duplication and division of cytoplasm and organelles, ultimately to produce two new daughter cells. Also, we can say that the cell cycle is an ordered series of events involving cell growth and cell division that produces two new daughter cells. Cells on the path of cell division proceed through a series of precisely timed and carefully regulated stages of growth, DNA replication, and division that produces two identical or clone cells. Now, what are the phases included in cell cycle? So, there are two main phases which are included in cell cycle: interphase and the mitotic phase. During interphase, the cell grows and DNA is replicated. During mitotic phase, the replicated DNA and cytoplasmic contents are separated. Ultimately, the cell divides. Yes, so here you can see that in G1, S and G2 phase, all these three are interphase. So interphase carries measure of the portion of the cell cycle. Then mitotic phase, which is of mitosis and cytokinesis, where formation of finally two new daughter cells will occur. Yes, so let me give you the idea that what exactly will happen in G1 phase. So it is the growth phase where ATP synthesis, preparation for S phase, protein synthesis and RNA synthesis will occur. In S phase, centriole will divide, histone protein synthesis will occur, which is basically DNA packaging proteins, and DNA replication will occur. In G2 phase, preparation for cell division, protein synthesis, spindle fiber formation, plasma membrane formation, ATP synthesis and RNA synthesis. Ultimately, mitotic phase where cell division will occur. Now, as I said that G1, S and G2 phase is actually a interphase which is preparative phase, which is further divided into G1 or gap 1 phase, S phase or synthetic phase, G2 or gap 2 phase. And cell division phase, which is mitotic phase, is further divided into two categories, which is karyokinesis, that is nuclear division or mitosis, let's say, and cytokinesis, which is division of cytoplasm. Now interphase, as I said that it is the preparation phase, it carries about 95% of the time of the overall cycle, and during this phase the cell undergoes normal growth processes and also prepares itself for cell division. In order for a cell to move from interphase into the mitotic phase, there are many external and internal conditions which cells have to met. So each of these things will be carried out during interphase, and it carries three phases as we discussed in the above slide, that G1 phase, S phase and G2 phase. Now interphase, as we said, is divided into three phases. What exactly happens in gap 1 phase? So in G1 phase, cell is going to prepare itself for S phase. For every phase, cell will prepare itself for the next stage. So in G1 phase, cell is going to prepare for S phase. What will happen in S phase? In S phase, there will be replication of DNA. Now, why do we need DNA replication? We need to understand this. See, if the cells divide, both the cells should get same information, or say complete information, right? And if we talk about human chromosome, there are 46 chromosomes. Now parent has 46 chromosome, then how can it give 46 chromosome to both the daughter cells? It cannot, right? Since the parent have only one pair of 23 chromosome, which is basically 46 chromosomes, so it cannot give to both the daughter cells. So in that case, it is necessary to make a copy of 46 chromosomes. Let's say photocopy must be done, right, in order to give 46 chromosome to each of the daughter cells. So this thing will happen in DNA replication, photocopy of 46 chromosomes. If the initial quantity of DNA in the cell is denoted by 2n, then after replication it will become 4n. Of course, because we are talking about photocopy, it will become double, so it will become 4n. However, the number of chromosome does not vary. If the number of chromosome during G1 phase was 2n, it will remain 2n at the end of S phase. Also, the centriole divides. See, here we are talking about animal cell in particular, so we will talk about centriole, that it divides into two centriole pairs in the cells which contain centriole. We know that centriole is not present in plant cell. Now G2 phase. During this phase, the RNA proteins and other macromolecules which are required for multiplication of cell organelles and for spindle formation and cell growth are produced as the cell prepares to go into the mitotic phase. That will happen in G2 phase. Also, I would like to say that we talked about S phase. Okay, so let me again repeat the same, that in G1 phase protein synthesis will increase, as I showed you in the figure that protein synthesis occurs. What are protein synthesis? Basically enzymes which are required for DNA synthesis. Also, I would like to here say that RNA synthesis will occur, ATP synthesis. Obviously energy is must, right, for all these kind of processes. Also, raw materials which are required for S phase will be synthesized in G1 phase. Now, due to all these processes, what will happen? The size of the cell will increase, or let's say that cell growth will take place. And then the cell enters to the S phase where DNA replication occurs, and ultimately after that we saw that G2 phase. So next up is M phase. What will happen in M phase? Now it is mitotic phase where mitosis will occur. This is the mitotic phase of the equational division, as the cell undergoes a complete reorganization. Now, why reorganization? Of course, to give birth to a progeny that has the same number of chromosomes as the parent cell. As I discussed that each of the daughter cell should get 46 number of chromosomes. Mitosis is the form of eukaryotic cell division that produce two daughter cells with the same genetic component as of the parent cell. It is the continuous process, and the other organelles are also divided equally by the process of cytokinesis, which is preceded by mitotic nuclear division, which is karyokinesis. When we say word karyokinesis, we are referring to mitotic nuclear division. And when we are saying cytokinesis, we are referring to cytoplasm division. Now, M phase, the mitotic phase, further we know that it is divided into four stages, namely prophase, metaphase, anaphase, and telophase. There is one more phase which is in between prophase and metaphase, which is not described in many of the reference books, but in some it is been described. Yes. So what happens in prophase? Chromosomes condense and becomes visible, nuclear envelope breaks down and nucleolus disappears. As I said that there is one more phase in between prophase and metaphase. It is prometaphase. What happens in prometaphase? Chromosomes continue to condense. Kinetochores appear at the centromere, which is the central part of the chromosome. Mitotic spindle microtubules attach to kinetochore and centrosomes move toward opposite pole. This all the process, all the stages you have already studied during your cell division. So I would not elaborate it. You already know that it occurs in four stages. Here one more is added, which is prometaphase, and at last cytokinesis, where animal cells, a cleavage furrow separates the daughter cells. Plant cells, a cell plate separates the daughter cell. Since we are talking about only animal cells, so we are concerned with the animal cell only. Next, cytokinesis or cytoplasmic division. What is cytokinesis? I have already given you the one line definition, that is division of cytoplasm between two daughter cells. Cytokinesis or cell motion in the second main stage of the mitotic phase, during which cell division is completed via the physical separation of cytoplasmic components into two daughter cells. Division is not complete until the cell components have been apportionated and completely separated into two daughter cells, which will happen only and only after cytokinesis, which is after cytoplasmic division. There is one more phase, which is G0 phase or resting phase. Cells in G0 phase are not actively preparing to divide. They are in resting stage. The cell is in a quiescent or inactive stage that occurs when cell exit the cell cycle. When cell is about to exit or left the cell cycle, during that time G0 phase is initiated. Some cells enter G0 temporarily until an external signal triggers the onset of G1. Other cells that never or rarely divide, such as mature cardiac muscle and nerve cells, remain in G0 permanently. Now, there are many such cells which undergo only one time in a cell cycle, and after that they does not divide. Now, what happened to such kind of cells? So such kind of cells become specialized for performing particular job. Such kind of cell are initiated or they undergo to G0 phase, which is resting phase. Yes, so this was all about cell cycle and content oriented, so you can easily write your answer from this video lecture session. Now, I shall see you in another guest lecture session with one more new concept. Till then, stay safe and keep watching Study with RK. Do like, share, and don't forget to subscribe the channel. Thank you.